Dear students, greetings to all. In this module, we'll be continuing with our ongoing chapter, Chapter 6, Molecular Basis of Inheritance. The search for genetic material, the experimental evidence so as to prove that DNA is the genetic material. The search for genetic material, that is, whether the genetic material is DNA, RNA, proteins, or a carbohydrate moiety, the first experimental evidence for that was provided by Frederick Griffith in 1928, and he used mice, and the bacterium Streptococcus pneumoniae was used to carry out the study. Streptococcus pneumoniae has two strands, the rough and the smooth strain. The rough strain, it is non-virulent, and it is rough because there is no mucus polysaccharide coat, and therefore it observes rough in appearance. Whereas smooth strain, it has the mucus polysaccharide coat. It is virulent, causes pneumonia, and the colonies, they appear smooth in nature. The experiment which was carried out by Griffith, the experimental animal that was used was mice, and in the first step, the R strain, the living R strain, was incorporated into mice. Because the R strain of Streptococcus pneumoniae is virulent, it led to pneumonia in mice, and the mice died because of that. The living R strain in the next step was incorporated in the mice, and the mice lives. The reason being R strain is non-virulent in nature. The S strain in the third step was heat killed and was incorporated into the mouse, and it was found that because the S strain was heat killed, it lost its property of virulence, and therefore the mice survived. When a mixing of the heat-killed S strain and the live R strain was carried out, and then the incorporation done in mice, the mice died. The reason being, something got incorporated, something got transferred from the heat-killed S strain into the live R strain that made live R strain virulent, similar to S strain. So that gave the clue to Griffith that there is some material which is acting as a genetic material and is getting transferred from the S strain and conferring the property of S strain into the R strain. The search for the genetic material, therefore, by these observations which he had carried out when he did the experiment on mice, by the infection with Streptococcus pneumoniae, he came to the conclusion that there is some material which is referred to as the transforming principle, which is getting transferred from the heat-killed S to the R strain, and that enabled the R strain to become virulent. That is because of transmission of some genetic material. The experimental evidence, the proof for the biochemical experimentation for that was carried out by the biochemical characterization of transforming principle was done by Oswald Avery, Colin MacLeod, Maclean McCarthy. So as to determine the biochemical nature of transforming principle in Griffith's experiments, they purified the biochemicals from the heat-killed S strain. The basic biochemicals that were extracted included the proteins DNA and the RNA in the three test tubes as can be seen in the diagrammatic representation. To these tubes from which the biochemicals were extracted from heat-killed S strain, the proteases was added. The DNases and the RNases were added to the tube which were containing protein, the protein, RNA, and the DNA. Later, to it was added the R cells. It was found that in the tube where the proteases and the RNases were added, and later to it the live R cells were incorporated, transformation occurred. Transformation occurred, thus proving that it is not the protein or the RNA which act as the genetic material, because the enzyme proteases broke down proteins, the enzyme RNases broke down RNA, and even after losing their structure, the transformation took place, the R cell became virulent, thus proving that protein and RNA, they are not the genetic material. But in the tube which contained the DNases, which broke down DNA, the genetic material, that is DNA, got broken down, and no transformation was observed in such tubes. The reason being, the DNA acts as the heredity material which helps to transfer the genetic information, and when it got lyased, there was the material got lost which could help in transmission of the strain from the virulent heat-killed S strain to the live R strain, making the live R cells virulent in nature. Thus, it was proved that DNA was the transforming principle. Hershey and Chase experiment, which is also referred to as the blender experiment. Hershey and Chase, they grew some bacteriophages. The bacteriophage, T2 bacteriophage, was infected to E. coli, that's Escherichia coli. They were grown separately on two different medium, one which contained radioactive phosphorus, that was P32, and one which was labeled with radioactive sulfur, that is S35. Phosphorus is a component of proteins and sulfur is a component of DNA. So the viruses grown in P32, they got radioactive DNA because DNA contains phosphorus, and the ones grown in 35 got radioactive protein because protein contains sulfur. The bacteriophage, that is T2 bacteriophage, was then infected onto the E. coli. The viruses, they attach to the bacterial cell wall with the help of the tail fibers. It was deciphered that when infection was carried out with the bacteriophage having the S35 labeled protein and the one having P32 labeled DNA. Firstly, the first step which was carried out, that is the infection. Later to it was blending and then centrifugation. When the bacteriophage, it infects the bacterial cell, only the genetic material gets incorporated and the protein coat, the capsid, is left in the outer medium. So when the experimentation was carried out, it was found out that the radioactivity in case of the one which was infected with S35, the radioactivity phage protein that was there in the liquid. In case of the one which was infected with P32, the radioactivity having the phage DNA, it was in the pellet. So the one which was radioactively labeled with P32 that got incorporated in the cell, that means that the DNA is the genetic material which gets incorporated in the cell, and that is the DNA is the material which carries the genetic information. Whereas the protein code, the one which was labeled with S32, it was found that the radioactivity was detected in the supernatant because sulfur did not get incorporated inside the bacterial cell, thus proving that DNA is the genetic material which helps in the transmission of characters from one generation to next, because protein did not enter into the bacterial cell, and it was found that radioactivity in case of the one labeled with S32, it was there in the supernatant, whereas the ones which was labeled with 32, phosphorus 32, it was detected in the cell because phosphorus is a component of DNA and DNA gets incorporated incorporated in the cell, thus proving that DNA is the genetic material. In this module, we discussed about the experimental evidences that helped to prove that DNA acts as the genetic material which helps in the transmission of heredity characters. Hopefully, you must have garnered all the concepts thoroughly. Thank you.